Okay, if you're watching this video, then you've presumably watched the one that I just made before about drawing cyclohexane chairs, because this structure here is the one that I drew. I've copied this over onto this next video, because what we're going to talk about here is what a ring flip is, how we can draw the chair after we've done our ring flip, and how that has an effect on the stability of the ring based on whatever substituents are present in the molecule. We're going to be talking about things called 1,3 diaxial interactions in this video. Okay, 1,3 diaxial interactions. Now, in order for us to actually do that, I need to make one of these hydrogens here, because right now I'm assuming all the bonds here are hydrogen. I'm going to make one of these a methyl group. So let's say I make this guy here a methyl, but actually I'm going to do it with two of them. I'm going to make this guy a methyl group 2. So I have one methyl group that's axial, one methyl group that's equatorial. Okay. Now, before we do anything, I want to see if you guys can figure out what two-dimensional representation this corresponds to. Notice there's the cyclohexane ring, there's the methyls on carbon 1 and 4. But when we show stereochemistry with these guys, we usually just show wedges or dashes. So on carbon number 1 here, if this is carbon 1, how would the methyl group be represented? Well, it's pointing up axially, but on these structures, axial and equatorial isn't something you can represent. All you can show is up or down. So there's your CH3. And also on carbon 4 here, this is also up, but bear in mind it's not. It isn't shown as either axial or equatorial because you can't show that here. So what we have here is 1,4-dimethyl cyclohexane, or more particularly cis 1,4-dimethyl cyclohexane. And for every different isomer of this, you're going to have a different chair structure. So let's just take a look at what we have here. So I want to take a look at 1,3 diaxial interactions on this ring before we do any ring flipping, okay? If you remember, the 1,3 diaxial interactions are energetic interactions that make your molecule less stable. Basically groups that are too close to each other, bumping into each other and saying, get out of my space. And for anything that's equatorial, you can see it's already pointing away from the ring, essentially in its own direction. So this equatorial group is not going to interfere with this one, and unless this one's particularly big, it's not going to interfere with this one either. It's the axial interactions that you want to be concerned about. So the way to figure this out is, well, first off, you need to know where your axial group is. So it's here, on carbon number 1. And you want to ask yourself, well, how many other carbons do I have that have axial bonds pointing straight up? Well, if you can see, there are 2 carbons, carbon 5 and carbon 3. And each of those is in a 1,3 relationship with this methyl group. On each of these carbons is a hydrogen. Okay, so what this means is, on each of these carbons, there's a hydrogen that's straight up an axial, just as there is on the methyl group. So what that means is this methyl group is actually a lot bigger than it looks, and the electron cloud around this methyl group starts to overlap with this hydrogen. And so what we get is this unfavorable interaction here. And we have one here as well. The hydrogens are too small to have interactions between themselves if they're 1,3 to each other. And as we said before, there is nothing going on with the equatorial methyl group because it's pointing away from the ring, away from everything else. Okay, so now we have that sorted out. Let's see what we can do with our ring here, okay? If you remember from when you looked at this previously, The carbons that we're interested in when we're doing a ring flip are the ones on each end of the molecule. I've numbered these 1 and 4. You may have numbered them something differently. I usually tend to number them 1 and 4, but it doesn't really matter. The thing you have to remember, and this is what models are particularly useful for, so you can have it in your hands and see it, is that when you flip your cyclohexane chair, you pull the side on the end that's up, you pull it down. You pull the side on the end that's down, and you pull it up. So what that means is carbon number 1 that was originally top right is still going to be on the right, but now it's going to be on the bottom. Carbon 4 that was originally bottom left is now still going to be on the left, but it's going to be up in the top. Now, here's the way to draw this out. What you want to do before you put any of your substituents or anything else on it is you want to be able to draw out something that is essentially the mirror image to these 6 bonds here. Alright? So what I'm going to do is I'm going to draw out my 2 central carbon-carbon bonds, but I'm going to draw them out opposite to the way they were. I'm going to draw them out opposite, kind of like so. I'm going to try not to mess it up. So we've got this, and we have this. Alright, I've drawn them at a slightly bigger angle, but oh well, my apologies. And then I'm going to take my blue bonds, draw them out like so. Okay, and hang on a minute, let me make that one a little bit wide. Let me see, let me take that blue one again, and let me make this black one just a little bit longer. Excuse me messing up here, just a little bit longer. Okay, I'm going to do my blue one here. Oh dear, bloody hell. I'm sure you can make yours prettier than mine, but on the other hand you'll be able to see exactly where you were drawing. It's the only thing that is a problem about doing these. Okay, and then we're going to join up our bonds. Alright, now I'll be perfectly honest, this is not the best representation, so what I'm going to try and do here, if I can, is I'm going to try and rotate. Oh, balls. That's not what I wanted to do. I guess I can't rotate the whole thing. Hang on a minute, let me see if I can. Yeah, talk amongst yourselves whilst I'm doing this. La-di-da-di-da-da. I guess I'm not going to be able to rotate it. Anyway, never mind. We can handle it. Okay, my apologies for this not being exactly opposite, but it's close-ish. Okay, before we do anything, let's put on our number locators, just so we know exactly what's what. You remember we said carbon 1 was originally up right, now it's going to be down right, it's going to be here. Carbon 4 was originally down left, and now it's up left, here. Carbons 2 and 3 were in the front of the molecule here. 1, 2, 3, 4 being the front of the molecule, and 4, 5, 6, 1 being the back. So therefore 2 and 3 are still in the front of the molecule. 2, 3. So therefore what we have here is 5 and 6. Okay. One thing about flipping your ring is that every group that was axial becomes equatorial. Every group that was equatorial becomes axial. Okay. But whatever was up stays up. It just switches from axial to equatorial. So let's start off with carbon 1. We originally had a group that was axial up and equatorial down. So what that means is we have 2 bonds now meeting pointing down, so our axial bond points down. And in fact we can go ahead and fill all these in, same as we did before on the previous video. Like we said before, the axial bonds are the easy ones. We've got to do the equatorial ones. So this was originally axial up, now it's going to be equatorial up. And if we follow our method, what we used last time, walk along here, we get to this bond, walk along here, get to this bond. You can actually follow that for all of these, and what you'll be able to do is to draw out all of these bonds in the right place, like so, like so. Well, let me correct that one. Now I've made a little bit of a mistake here, in that my these two red bonds are a little too horizontal. That's yeah, just like that one is. Isn't supposed to be a banana. That was, oh dear, now I'm deleting my whole structure. Hang on. Go back here. Let me fill in everything else. That's the way it's supposed to be. A little bit banged up, but what the hell. Okay, that's better. And then we have just one more bond to put on. And that's our axial hydrogen there. Okay. So what we need to figure out is, where are our two methyl groups? Well, they're on carbons 1 and 4, and they haven't moved. Initially, the methyl on carbon 1 was up and axial, so it's still going to be up, but now it's up and equatorial. Remember what we said before about equatorial groups? They don't interact with anything else because they're in their own direction away from the ring. Groups that were equatorial become axial, so this is equatorial up, this is now axial up. So the other group that we're particularly concerned about is the one that's axial, which is the methyl on carbon 4. So, with the methyl on carbon 4, we have to find which hydrogens are interacting with it. So we have to do what we did before. Which hydrogens or which bonds are also axial up? And it's the ones on 2 and 6. Remember that when we talk about 1,3 diaxial interactions, we're talking about what groups are on position number 3 relative to this one. Don't worry about the actual numbering here. If you can go 1, 2, 3, and both are axial, those groups will interact in a 1,3 diaxial interaction. Just as you can do 1, 2, 3. So these two will interact, and these two will interact. Remember we said before, hydrogens are too small to interact with each other. So how many diaxial interactions do we have on this conformation? Well, it was 2. And how many do we have for this conformation? Well, in this case, it's also 2. So what that means is neither conformation is more stable than the other, so you get approximately a 50-50 split. If the group that's a substituent changes, that's going to change everything, because some groups are bigger than others. Some groups have more of an interaction with axial hydrogens. And if you had something like, for example, 1,2 dimethyl cyclohexane or 1,4 dimethyl cyclohexane, sorry, the 1,4 is what we're doing. We could have 1,3. Or you could have ethyl groups, propyl groups, T-butyl groups, chlorine, anything like that. The principle behind what you do is exactly the same no matter what the substituent is. So that's how you flip a ring, that's how you treat your substituents, and that's how you figure out which is more stable.